In this section, we will talk about anchor amino acid, allele-specific motifs, and T cell activation. MHC class II-dependent antigen processing exogenous antigens, including foreign molecules or microorganisms, must be internalized, digested into peptide fragments, and bound to the peptide binding groove of MHC molecules before antigen-specific CD4 plus T cells recognize them. Through receptor-mediated endocytosis or phagocytosis, exogenous antigens are conveyed into endosomal vesicles. These develop from parts of the internalized cell membrane and have a neutral pH, so acidic proteases are inactive. Within hours, the pH of the endosome decreases, and internalized proteins are digested by cystine proteases, called cathepsins. The endosomes then fuse with vesicles containing MHC class II molecules. Recently, synthesized transmembrane proteins, such as MHC molecules, are translocated from the cytosol to the endoplasmic reticulum, where they are folded and assembled correctly. They'd first occur as dimers of alpha and beta chains. A third chain, the gamma or invariant chain, binds non-covalently to MHC2 alpha-beta heterodimer, blocking its binding site. This prevents the binding of endogenously synthesized peptides, which are abundant in the endoplasmic reticulum. The part of the invariant chain that blocks the groove of the alpha-beta heterodimer is called class II-associated invariant chain peptide, or CLIP. The protein, calnexin, retains the complex within the endoplasmic reticulum until assembly is correct, then dissociates and allows the complex of MHC2 and invariant chain to leave the endoplasmic reticulum through small coatomer-coated vesicles. These fuse with the Golgi apparatus on dissociation of the coatomer-coated protein. The invariant chain targets the MHC complexes to transport vesicles of the trans-Golgi network, which traffic to endosomal vesicles. The invariant chain is cleaved, leaving only the small clip fragment bound to the binding site of the MHC2 heterodimer. The compartment where fusion of the transport vesicles with the endosomes occurs is called MHC class II compartment. An MHC class II-like molecule called HLA-DM induces release of the CLIP peptide from the binding groove of the MHC and stabilizes the empty MHC2 heterodimer until an appropriate exogenous peptide is bound. Finally, HLA-DM dissociates and the peptide MHC2 complex is transported to the cell membrane. Peptides that were unable to bind to MHC molecules are degraded in lysosomes. Now let's discuss the anchor amino acids in MHC2 peptides. The peptide binding groove of MHC class II molecules is open at both ends. So the size of peptides that can bind ranges typically from 12 to 24 amino acid residues. Both chains of the MHC2 molecule interact with the side chains of the peptide and determine binding affinity. Each class II allele has different side chain contacts, allowing only peptides with certain amino acids to bind into particular key positions or anchor positions. The anchor position closest to the amino terminus, which is position 1, accepts only aromatic or large aliphatic amino acids. This is essential for high-affinity peptide binding. Other important, although less critical anchors are the amino acids at peptide position 4, 6, and 9. On the other hand, side chains of certain amino acids can interfere with peptide binding, called as inhibitory amino acids. Anchor positions are the most important contact sites of the peptide with the MHC2 molecule. So it is not surprising that the same key positions are critical for binding inhibition. Now, we will discuss the amino acids that favor or are anchor amino acids and inhibitory amino acid. So, at position 1, phenylalanine, isoleucine, leucine, methionine, tryptophan, and tyrosine are anchor amino acids. At position 4, aspartate, methionine, glutamine, and serine are anchor amino acids. And inhibitory amino acids for position 4 are glycine, lysine, proline, arginine, tryptophan, and tyrosine. At position 6, anchor amino acids are serine, threonine, and valine. But inhibitory amino acid at position 6 are glutamine, phenylalanine, glycine, histidine, lysine, leucine, methionine, and tyrosine. At position 9, anchor amino acid is serine. But inhibitory amino acids of position 9 are aspartic acid, glutamine, leucine, asparagine, proline. The clip peptide of the invariant chain can bind promiscuously to different MHC2 alleles. It has methionine at position 1 and side chains that are accepted by all DR alleles in positions 2 to 9. Computer programs can now predict binding motifs for a particular HLA-DR allele from the protein sequence. Now let's discuss T cell activation through MHC2. The peptide MHC class II complex can activate CD4 T cells and induce the proliferation and secretion of various cytokines such as interleukin-2, interferon gamma, released by CD4, cause release of tumor necrosis factor alpha by the antigen-presenting cell, plays an important role in this process. It leads to the formation of oxygen radicals that are able to kill intracellular microorganisms. Furthermore, cell-presenting antigen cause CD4 cell activation, and activated CD4 T cells release interleukin-4 and interleukin-6 that can stimulate B cells to release antibodies and kill antigen. Both mechanisms are directed at the elimination of extracellular pathogens. This is all about anchor amino acid, allele-specific motifs, and T cell activation. This was all. We will discuss this T cell activation in detail in our upcoming sessions in this video. In our next section, we will talk about the molecules involved in interactions between T cell and antigen-presenting cells.